Okay, everybody. Hello, I'm Brian, and this is AAMI News and Conversation. We're here at day 3 of the AAMI FDA neXus conference here in Washington, D.C. And I'm here today with Dr. Julian Goldman. And Julian, why don't you tell us a little bit about what you do, who you are, your title, and then we'll talk about the session that just had a really good attendance rate, and it seemed like there was a lot of fascination with it.
Sure. Happy to be here with you today, here at the AAMI neXus conference. My name is Julian Goldman. I'm an anesthesiologist at the Massachusetts General Hospital and medical director of Biomedical Engineering for the Mass General Brigham health system. Also, I lead a research program on medical device interoperability and cybersecurity with an emphasis on smart and autonomous medical systems, which may connect with some of the things that we're talking about today.
So that session that I mentioned just earlier had a lot of focus on pulse oximeters. And you had mentioned that that focus has kind of been almost like a building wave. There's a lot of interest, a lot of standards work, a lot of regulation surrounding this. Why? What's going on there?
Well, it's really quite interesting. Pulse oximeters have been around for quite a while. Initially, they were used in anesthesiology, take care of patients intraoperatively, and they branched out slowly initially, but then they caught on like wildfire, and especially during the pandemic. As the awareness increased that there could be silent hypoxemia with COVID infection, that people at home, they may feel not great, but in fact they could be in some danger because their oxygen level might be much lower than they appreciated. That really increased the interest in using pulse oximetry. So that increase occurred out of the hospital, in the hospital, everywhere, globally.
Yeah.
And so there was a tremendous increase in awareness of the benefit of pulse oximetry. Suddenly there were people using pulse oximeters that were not medically trained, and they were using them oftentimes incorrectly, or they weren't sure how to interpret the information. New lower cost pulse oximeters were showing up on the market, online, sometimes for as little as $20, $30, $40. And there were many reviews online by people that didn't know very much saying, well, I tested this pulse oximeter in the doctor's office and it read the same as the medical pulse oximeter, so it must be correct. But of course, reading the same number when someone is healthy and sitting in the doctor's office is not the same as it tracking a desaturation in someone who's sick and may also have poor perfusion in their fingers, poor peripheral perfusion. So there was a tremendous increase in interest in pulse oximetry, and that occurred with some other kind of interesting developments that just happened to occur in the same timeline. And the other big development has been in the last decade or so, the increased access to patient information in medical records. It's much easier to mine medical records and to gather large quantities of data.
Right. Importantly, these are devices, these are patient monitoring devices.
Yeah.
So that plugs right into patient data, right? It's providing new information all the time.
And typically in many hospitals it's acquired either manually or automatically as part of the medical record. So in 2020, there was a letter published in the New England Journal of Medicine by Dr. Schoeding and team, and they looked at a large number of patients, and what they found was that they identified that in patients with darker skin color, or actually they self-identified as black or people of color, that the data seemed to show that their displayed oxygen saturation on their pulse oximeters that was located in the electronic medical record was a little bit higher than the value obtained in a blood sample that was drawn and sent to the lab. They looked at these numbers and they said, well, pulse oximeters are never perfectly accurate. You just don't achieve that with 2 wavelengths of light on a fingertip compared to a blood sample. But it seemed like there was a subset of patients, some were light-skinned, but somewhat more of them had darker skin pigmentation based upon really self-identified color, that had a greater difference between the value in the record and their actual oxygen saturation. And the problem is that the pulse oximeter was reading higher than the blood level, meaning the error was in overreading the oxygen saturation. So if we think a patient's oxygen saturation is higher than it really is, then they may not receive the therapy that they need. They may not receive as much oxygen supplementation or other therapy, and it may not be recognized that they are as sick as they are. So that's the concern of overreading, or what's called bias in the measurement between the measured saturation with a pulse oximeter and the actual saturation.
Normally, since we're talking about these devices, and as you said, there became more of them, there was a lot of attention on them, I can imagine that the design of these devices can vary, what they can perform, their efficacy might vary pretty widely. But at the end of the day, it's a tiny little thing. It just sits on your finger normally, right?
Well, you know, this is a typical inexpensive online purchase, right? Yeah. Pulse oximeter. And they're wonderful. This one, I'm not even sure this is actually a medical device. I'm not sure this one is a true pulse oximeter because it can be difficult when buying something online to know whether it meets the criterion set by the FDA that this is a medical device. If it is, it should have been, according to FDA requirements, should have been tested in the laboratory on, and the way the testing is done is determined by a standard, by an ISO/IEC standard 80601-2-61.
This is an international standard
recognized by the FDA. The latest version was published in 2017, and it's currently being actively updated. And the standard describes how to test pulse oximeters in the lab. The way that's done is with healthy adult volunteers, and those volunteers are given a mixture of a low oxygen concentration to drive their saturation to a low value. That's why they have to be healthy and they have to be volunteers. And when that testing is done, the blood sample is analyzed with a co-oximeter, which is an instrument designed to truly accurately measure oxygen saturation, and that's compared with the pulse oximetry measurements. So this is the kind of testing that's been done for some time. And when you buy a device that is a medical pulse oximeter, so a medical device that meets FDA requirements to be called a pulse oximeter, that validation is done. And oftentimes one isn't sure, and you have to kind of track the information and see whether this meets the criterion. So that's part of the system, is understanding there are requirements in the standard, and the standard doesn't require this clinical testing. Clinical is a tricky word because this is not testing in a clinical environment. It's being tested in a laboratory on healthy volunteers.
As you pointed out, these are not patients.
They're not patients. So that the standard describes how to perform the testing, why to perform the testing, the test conditions. The FDA describes the FDA to clear a device as a Class 2 medical device states you should, it doesn't say you have to follow the standard, but that's typically the easiest way. And the FDA states you should perform this kind of validation testing. And what was added to the testing some time ago was actually including subjects with darker skin pigmentation as well as those with lighter skin pigmentation, which was added by the FDA a number of years ago before the recent focus, the recent interest on concerns about performance in darkly pigmented patients.
Now that's interesting because it also tells me that these devices, our understanding of the technology, our understanding of the science is changing.
It is.
The devices are going to change with it. Does that also mean then that the regulatory approach needs to change, or the standards are going to change with it?
Well, and that's a very good question. Pulse oximetry kind of made its big splash decades ago, right? It was widely adopted and has become essentially a standard of care in many environments. And then there have been incremental improvements in performance, reductions of cost, miniaturization, and so forth, like as we see here. But really, the last few years, and I think it's that alignment of the fact that the ISO/IEC standard is in an update cycle. Concerns were raised initially in the letter that I mentioned by Dr. Schoeding and team, but that was followed by many other publications and a lot more recent work, including some funded by the FDA, to dig deeper into understanding whether these issues are common, are they pervasive, how significant are potential errors in pulse oximetry measurement, in which patient subsets does it matter. So the FDA has been digging deep into this at the same time as there's been increased interest and need by the public and by the manufacturers. And the FDA held a panel on pulse oximetry. They held several, but the most recent was February 2nd of this year. to discuss some of these issues and to tease out what might be causing, in what settings and in what patients might there be an increase in the measurement bias. It seems like the information showing up from the scientific and clinical research community is that it's probably a constellation of things occurring, but really it's not proven yet what the underlying mechanism is. But it looks like it's probably not only is it dark skin pigmentation, which, you know, a thick digit and a digit that's dark, both of those things decrease the amount of light penetration, and that could affect the signal to noise ratio, with the signal being the pulsatile signal that we're interested in measuring, pulsatile pulse oximeter, so the pulse.
And for those who don't know, you mentioned light penetration. So how does this device work then? Is there a beam inside the device?
Yeah, so basically pretty much all pulse oximeters have two LEDs, so two lights. They are typically applied on the nail side if it's a finger pulse oximeter, and one single photodetector on the palm side of the finger. And the two LEDs, the red and the infrared LEDs, cycle on and off because there's only one photodetector. So the photodetector knows, the system knows, okay, that now the red LED is on, so I'm measuring the amount of red light coming through. Next, the infrared coming through, and then there's a period where there's no light, and it can detect any ambient light that's coming through. So the calculation is made by cycling the red and the infrared LEDs.
That's so fascinating, because at the end of the day, and correct me if I'm wrong here, but it almost seems like this device is almost the ideal case study for how to approach, I guess, regulations and standards for devices that maybe the basic build of it is not going to change much, but our understanding of application for patients, our approach to validating it might need to change.
Well, actually, everything might change. So it's possible that adding more LEDs, more wavelengths, could improve the performance, but also increase the cost. It's possible that subtle changes in the design, not only possible, but manufacturers have shown that small changes in the design of a pulse oximeter can influence its performance. Because not all the light that comes out of those LEDs passes to the photodetector. It spreads, light goes where it wants. Also, the way that the oximeter fits on the finger, if it rotates a little bit, if it slips off and on, and so forth, that could matter greatly, and therefore the construction and its fit to the finger could matter in its performance and its accuracy. So when you're looking at these large data sets from a hospital, and remember they didn't use necessarily these devices, they used probably a lot more, well, they used those that you find in the hospital, which are often adhesive and stay in place. Sometimes they clip on, but they're not as heavy. The batteries in here make it heavier and it slides. Knowing whether something is fit perfectly is very hard to know. It's hard to know in a clinical data set. Also, the thing that we know can influence or affect or degrade pulse oximetry performance is poor peripheral perfusion. So if someone is cold, the fingers are cold, their perfusion is worse. If you press on the nail bed, anyone can do this and look at your nail and see how long it takes for the blood to return. If it takes, say, 10 or 12 seconds, your hands are probably cold and you have poor perfusion. If it happens in 2 or 3 seconds, probably warm and normal perfusion. Well, poor perfusion means poor pulsatile signal that the pulse oximeter has to detect. So poor peripheral perfusion provides less of a signal. So if someone is cold and maybe shivering and they have poor peripheral perfusion and maybe they have large fat fingers and maybe they have a lot of dark skin pigmentation, those are all factors that can challenge the measurement. But we don't know exactly why there is increased bias in some patient subsets. There are light-skinned patients in clinical data sets that also have an increased bias. So it isn't necessarily just skin pigmentation. And it probably is multifactorial, and as we know in life, if it's multifactorial and if you can't use another instrument because these are patients in hospital settings, that's where the data is from, it makes it very difficult to tease out the factors and then attribute the weighting of what those factors, how those affect the performance. So I'm actually very gratified that so many people are working on this. Not only the FDA with its resources and expertise, manufacturers all over the globe, the meetings that we've held in the Pulse Oximetry Standards Committee just show the intensity of interest. Clinical researchers are investigating both in the lab and in clinical environments. Experts in optics and statistics. There are new measurement techniques. There are, for example, optical techniques to measure skin color that are going to be advocated and will be included in the, at least they're in the draft standard. So there'll be a method to measure skin color and include that in a data set, for example. And whether that's an electronic instrument that measures color or a color card that can be held up to the patient to see their skin color. There are so many questions. You know, what about the skin color at the measurement site, which might be the fingertip? But when we look at people, we see their face, and someone could have a dark face and a very light fingertip. Mm-hmm. So is it really the color that's causing this performance issue? Or are there other factors that are occurring? Do we all have slightly different vascular beds for some reason? So we've opened Pandora's box, but we can't just wait and sit back and hope to study this for 20 years.
Right.
It has to be addressed.
There's a need for it now.
It's a need now, and you know, we'll I'm sure the community will iteratively refine what we learn and then what impact it has on pulse oximetry.
I've learned a lot just hearing from you briefly today, and I wanted to wrap things up asking if your lab is focusing on anything in particular right now.
Well, we are addressing some facets of pulse oximetry and of medical devices.
Right.
Now, pulse oximetry data is used typically manually. We look at the number as clinicians, make a clinical decision, or we ask patients to track it at home, let's say. But pulse oximetry data is also part of systems that are becoming more automated or have been automated for decades. For example, it's possible to control oxygen delivery, to conserve oxygen by monitoring pulse oximetry and automating that in a closed loop fashion. There's been a lot of advance in closed loop control. The FDA released guidance in September or October of 2023 on closed loop control. There's an international standard on physiologic closed loop control. It's IEC standard 60601-1-10. That's also has been revised. And to address this need to have smarter systems, our lab, our program at Mass General Hospital stood up a program. It's actually a collaborative community on smart and autonomous medical systems. And pulse oximetry becomes a key component of many smart and autonomous medical systems. So that collaborative community is being stood up right now, and I expect that everyone will be able to learn more about it in a few months as we move forward.
Fantastic.
Yeah.
This has been very enlightening. It's really fascinating to watch almost what seems like a revolution in what is a technology that's been around for quite a long time.
Right. It is interesting. You know, suddenly a number of things aligned, and it's going to potentially cause a revolution in how we use and how we see pulse oximetry, how it performs, and every facet, including how we train in its use. And hopefully pulse oximeters will become smarter instruments as well, and help us to use them more correctly.
Something to look forward to.
Yeah.
Well, thank you so much again for sitting down and talking to us, Dr. Goldman, and enjoy the rest of neXus.
Thank you. I appreciate the opportunity to speak with you today. Thank you.